My topic is regulation of gene expression, induction and repression systems in the Opron model. Induction and repression. Certain microorganisms could change their enzymic machinery under the influence of specific substrates. This phenomena is called as enzyme induction. The substrate whose introduction into the growth medium specifically increases the amount of an enzyme are called as inducers. Their corresponding enzymes are called as inducible enzymes, and the genetic system responsible for the synthesis of increase of such an enzymes are called as inducible systems. One of the best studied systems is beta-galactosidase, the enzyme that catalyzes the hydrolysis of lactose into galactose and glucose in E. coli. The enzyme is produced only when lactose is present in the medium. In the absence of lactose, enzyme synthesis virtually stops. It has been demonstrated that inducer of the enzyme and the substrate on which it acts may be or may not be identical. The opposite phenomena is called enzyme repression, where the end product of a biosynthetic sequence repressed the synthesis of enzymes involved. The end products whose present in the medium checks the synthesis of an enzyme are called as co-repressors. Their corresponding enzymes are called as repressible enzymes. The Opron model. The famous Opron was proposed by F. Jacob in 1961 in order to explain the induction or repression of beta-galactase enzyme synthesis in E. coli. Regulators may or may be located close to the genes which they regulate. Operators are always located close to and in fact may be part of the one of the structural genes they control. The operators and its structural genes constitute an opron. This opron is generated can be represented as mechanism of opron model. In inducible system, the presence of inducers is an essential feature for the synthesis of inducible enzymes. In the absence of an inducer, the system does not synthesize the enzyme. In case of E. coli, opron model regulates the synthesis of beta-galactase do not function, and because of the presence of repressors in the cells of activity of genes is checked. In an inducible system, the active repressors in the absence of an inducer attached with the operator gene. This preventing DNA transcription of an inducer is present. It combines with the active repressors, returning the repressors inactive. The inactive repressors is enabled to attach with operator gene. This permits the structural gene to be described. In repressible system, the repressors in the absence of co-repressors does not attach with the operator gene, this permitting DNA transcription. If a co-repressor is present, the repressor systems combine with it to form a co-repressors complex which blocks the operator gene. This is the DNA transcription does not take place. Mutation in controlling genes and protection of consecutive strains. The regulator and operator genes are called as controlling genes. It is not a well-known fact that each repressor blocks the synthesis of one or more proteins, and like all other proteins, repressors are coded by chromosomal DNA. The genes which code for them are called as regulatory genes. Thank you.